Hey, this is Rochelle. Welcome to another Study With Me video, Microbiology edition, where you don't just watch me study, but we study together. So in this video, we'll be studying transcription and translation. But before we get started, feel free to hit that subscribe button and that little bell right next to it so you'll get notified of when my videos will come up next. Also, if you have any questions or comments, put them in the comment section below. I also have a microbiology study program that is designed to be interactive and repetitive so that you'll remember the material better. I go more in depth. If you want information on that, my email is in the description box below. Feel free to email me. So let's get studying. Transcription. It is the synthesis of complementary strand of RNA from DNA template. So here I have a coding strand of DNA from 3' to 5'. Now, as usual in the DNA replication, we have our two enzymes. It's topoisomerase, which straightens the DNA helix, and we have helicase that unwinds the double helix and separates the two strands. Now, in transcription, there are three phases: initiation, elongation, and termination. First up, in initiation, we have our RNA polymerase that attaches itself to the promoter. And the promoter is a small section of the DNA of the coding strand. So we have our RNA polymerase that binds to this code, this promoter site. Let's put promoter here. Promoter site. Next, we have elongation. Now, the RNA polymerase moves from 3' to 5' along the DNA, the coding strand. As it moves along, it picks up nucleotides, and these nucleotides are A and U, G, and C. So we have our nucleotides, which connect to each other to make our messenger RNA. Now, this messenger RNA reads 5' to 3'. Now, eventually the RNA polymerase will stop, and it will stop at the terminator site. And this is our last phase, termination. All right, let's go over this. We have our coding strand of the DNA that's 3' to 5'. In our first phase of transcription, we have initiation. Initiation is the RNA polymerase binding to the promoter site. Now, in elongation, our second phase, RNA polymerase moves along the DNA coding strand from 3' to 5', synthesizing messenger RNA using nucleotides A, U, and G, C. Eventually, the RNA polymerase will hit a terminator site. and it will stop making messenger RNA. Now, RNA polymerase will synthesize the messenger RNA from 5' to 3'. All right, we're going to do this again. So here we have our DNA. Now our DNA has been straightened by topoisomerase, unwound, and the two strands divided by helicase, leaving us with our coding strand. Now our first phase of transcription is initiation. And what happens in initiation is that the RNA polymerase binds to the promoter site. Our second phase is elongation. And this is where our RNA polymerase moves along the DNA coding strand, moving from 3' to 5', picking up nucleotides, RNA nucleotides. Eventually, the RNA polymerase will stop, and this is the termination phase. Now, it stops at the terminator site. Once it stops, it leaves us with our final messenger RNA reading from 5' to 3'. And this is transcription. First up, I want you to tell me what this is. And you can place it here. What is the first step of transcription? I need the name and the process. What is the second step in transcription? The name and the process. What is the third stage in transcription? The name and the process. And what is this enzyme that's involved? All right, let's fill it in. So here we have our DNA strand. It's our coding strand. Our first phase or stage is initiation. And that is when the RNA polymerase attaches itself to the promoter site. Our next phase is elongation. So our RNA polymerase moves along the coding strand of DNA in the 3' to 5' direction, picking up our RNA nucleotides. Eventually, our RNA polymerase will stop, and it will stop at the terminator site. And this is our last phase, termination. Now we're left with our messenger RNA that reads 5' to 3'. And this is transcription. Translation is protein synthesis. Now, protein synthesis is the decoding of messenger RNA and converting it to protein. Now, here we have our ribosome. It is the site where protein synthesis occurs. So we have ribosome. We need our messenger RNA. Now our messenger RNA would typically go here, but I can't write on this, so we're going to have our messenger RNA. Now that we have our messenger RNA, let me label that messenger RNA. Every group of 3 nucleotides is a codon. And the codon codes for a protein. Okay. Here we have our transfer RNA. We have our amino acid up top and our anticodon on the bottom. It is complementary to a specific codon. So transfer RNA comes in, reads the codon, and the amino acid is left. Another transfer RNA comes in, reads the codon, and leaves the amino acid. Another transfer RNA comes in, reads the codon, and leaves its amino acid. Now we have our amino acids that's linked by peptide bonds. And this is a polypeptide chain. There is something that I do want to add. We have a start codon, which is AUG. And we have our end codons. And this is how I remember them. So I do A across. Oh, I'm running out of space. and this is our end codons, and this is how I remember it. U, A across, U, down, U, A diagonal going up, U, A diagonal going down. The blank spots, you fill it with G. And those are your codons that code for the end. Okay, translation. We have our ribosome. It is the site of protein synthesis. We have our messenger RNA. Our messenger RNA has codons. Our start codon is AUG. Our end remember, it's UAA across, U down, UAA going up and up diagonal, and UAA going down diagonal. And the missing spots, you fill it with a G. And those are our stop codons. This one is start. Okay. Our transfer RNA will come in and bind to a codon. The transfer RNA has an anticodon that's complementary to the codon. The transfer RNA also has an amino acid. Whereas codon codes for this amino acid. Another transfer RNA will come along that's coded for this particular codon. It has anticodon that's complementary to this codon and amino acid. The codon codes for this particular amino acid. Another transfer RNA will come through the ribosome and will bind to this codon. This transfer RNA has an anticodon that's complementary to this particular codon. This transfer RNA has an amino acid. This codon codes for this particular amino acid. Now these amino acids are bound together by peptide bonds, making this a polypeptide. Okay, in this part of the video, I'm going to test your knowledge on translation. So here we go. What is this? It's a ribosome. And it is the site of protein synthesis. What is our start codon and what are the end codons? Our start is AUG. Our end, remember, UAA across, UUU down, UAA diagonal going up, and UAA diagonal going down. The missing spots, you're going to fill it up with G, and this will give us our end codons. So we have our transfer RNA that has our anticodon, and what else linked to it? Amino acid. We have our messenger RNA, and our messenger RNA consists of codons, so what happens after this? The transfer RNA finds a codon. The anticodon is complementary to the codon. The transfer RNA has the amino acid at the top. The codon codes for the amino acid. Now this happens until the end codon is reached, and we have our chain of amino acids. Now, what is the bond that links these amino acids together? Peptide bond. And this whole chain is called a polypeptide. So that's translation.